Κορινά. Ναι. Μπες εσύ. Κορινά. Τώρα δεν πήγατε. Good morning, Joe.
Good morning, Konstantinos. Nice to see you.
Good to see you.
Good morning.
Can you hear me?
I can hear you well.
Yes.
Cannot hear you, I think, for some reason. Can you hear me?
Can hear you well.
Yes.
Okay. I can hear you also. That's good. I can see you also, and you can see me. So let's wait 2-3 minutes.
Hello.
Hello. Hello, Michael.
Hello, everybody.
Hello, Andrea. Hello, Ligia.
Hey, good morning. morning.
Morning.
Okay, good morning. Good morning, everybody.
Good morning.
I'm glad to see familiar faces on the webinar today. I hope everybody is doing great. And today it is a great pleasure for me. My name is Konstantinos Makris from the Cyprus International Institute for Environmental Public Health here in Cyprus, together with great colleagues from Cyprus and Norway, that we will try to explore some interesting facts and findings about hydrocarbon exposures and human health, with particular emphasis on night shift workers, either here in Cyprus or in Norway and in Bulgaria. So we're going to have, I would say, a pretty wide scenery of settings, worker populations, and study methodological approaches that I believe that all this interesting cocktail will give a very, I would say, interesting view of the topic on night shift work. And night shift work, as you all are aware of, it's becoming more and more important and common in nowadays life. So it provides on one hand the great flexibility. On the other hand, it might have some important ramifications for human health, because there is increasing evidence, particularly, let's say, with carcinogenic effects of night shift work that are accumulating in the global literature, along with other health outcomes of interest. So today, with a great set of plenary speakers, we're going to explore these aspects in more detail, both on the exposure side and also on the epi side. I would say more about the topic, but I think it's time to let our speakers do the job. So with no further ado, I'm going to call the first speaker of today's webinar. The first speaker of today's webinar is Dr. Joe Stenheim. He is a senior researcher in the section of epidemiology and prevention within the Department of Research at the Cancer Registry of Norway. And he's going to talk to us about the lessons learned and directions for future studies with respect to hydrocarbon exposures and cancer risk in the very important Norwegian offshore petroleum workers cohort that his group is coordinating. And he has actually spent lots of years on trying to set this up. So we know the hurdles that their team went through to accomplish this, and we would like to congratulate you on this very important initiative, and we would like to hear more about this. Joe, the floor is yours.
Thank you so much, Konstantinos. It's a pleasure to be able to be here and join the webinar, which I think is really interesting in terms of learning more about the mechanistic side, but also on the public health more. Let's say, higher level side in terms of data granularity. So I think we can combine very interesting angles of attacking the same research question, which I find very, very interesting. Okay, so I tried to share my screen. Let's see here. I hope this is the correct screen. Okay, and then
Yes.
I try to go presenter mode.
Exactly.
Okay, so I'll start. So as Konstantinos just said, I will try to give a brief account of the studies we have been doing in the latest years in this cohort, give a short introduction to the group working on these studies, and then a short introduction to the cohort, and then some results and some thoughts about forthcoming studies that we're planning the coming years. So this is our group, a recent picture just a few weeks ago. We're expanding, and we try to use and exploit the cohort, and also a new cohort that we are currently establishing as best as we can. And we also have a strong connection to the University of Oslo, where we are able to study other outcomes than cancer as well. So we are kind of a joint group of two institutions, both the Cancer Registry at the hospital, but also at the University of Oslo and the Department of Biostatistics. So currently we have these projects going on. We have the what we call the Norwegian Offshore Petroleum Workers Project, which is has 3 work packages. 1 PhD, the red line there, Philip, he completed his PhD last year. We have our postdoc Leon, who's working on respiratory cancers. I think Leon and Nita and Nikki joined us via the one of our group rooms, so they're joining us today as well. And then we have Nita, who is currently at the very final stage of her PhD. She's looking at urothelial cancers, bladder and kidney primarily. And then we have Ronnie, who's looking at lung and colorectal cancers. And all of this is connected to hydrocarbons, all of these studies mainly. The respiratory cancers is also connected to dusts and fibers. Then we have Besad, who many of you know already, who joined our group this June. And he will look at benzene, night shift work, and lymphohematopoietic cancers. And then we will try to use both the old cohort and the new cohort. I will talk more about the cohorts later. And then we have Nikki, who will join us. She's part of the group today, but she will join us in terms of looking specifically at offshore workers after Christmas. And then looking at CVD, cardiovascular disease, and mortality studies. So this is our ongoing activities. But today I will focus mostly on cancer and hydrocarbons and shift work. So a little bit first about the cohort. It was established in 1998 by questionnaires. So this is a questionnaire-based cohort, which has some advantages, but also some important drawbacks. It's not register-based, which means that we only have persons who were alive and resident in Norway at the time being in 1998, so that those who died from disease or who were emigrated for other reasons, we don't have them in the cohort, which might lead to some selections. They reported, so that's a typical drawback. More of the advantages of the cohort is that when you have the questionnaire data, as opposed to the registered-based data, you can have much more data. You can get quite a lot of data from a questionnaire. So we have educational level, complete work history for most of them. Also outside offshore work, we have some leisure time activities, and we have lifestyle factors like physical activity, anthropometry, alcohol consumption, tobacco consumption, and so on. So we started out with a list of about 61,000 persons, and then we removed those who were dead or emigrated, and we had a list of 57,000 persons. Out of those 57,000, we have the 22,000 who did not respond. We have the 7,000 who responded that they were never offshore, and then we were left with the green pie, which was the 28,000 that actually responded and then were defined as the Norwegian Offshore Petroleum Workers cohort. So we have a cohort of 28,000 persons who worked offshore from the very beginning of the Norwegian industry era, from the mid-60s until 1998. Yes. So the reason why we went about this as survey-based was that this list that I talked about of 61,000, it was not existing anywhere. So it was compiled based on likelihood of being an offshore worker. So we went to the county physician in Rogaland, which is the county of Stavanger, the main oil petroleum city in Norway. There we got some records. We went to the companies, oil companies, and we got some records from them. And we went to the maritime schools and diving schools, and we got some records from them. So it was kind of just a very scattered map of sources that we explored to compile this list. So that's how it was started. This was reported in 2001. That was the first report about the cohort establishment. It was done by Strand and Andersen, two former researchers here at the Cancer Registry, and that was written up in this report. Then in 2021, a couple of years ago, we published a cohort profile in the International Journal of Epidemiology to have that as a kind of a hallmark publication that we can refer to in terms of how it was established, some main characteristics of the cohort, and also some main findings up until the time of 2021. Yeah, so briefly from this publication, 4,000 cancer cases were identified by 2017. I think now we are closer to 5,000 with the most recent linkage, it's kind of unique because it's only offshore petroleum workers. There are a couple of other cohorts in the world with petroleum workers, but they are not only offshore workers. So we have the Australian Health Watch cohort. It contains both offshore and land-based workers, and we have some UK workers, and we also have some Canadian workers at the Canadian cohort. So there are different comparable cohorts, but our is, as far as we know, the only one with a pure offshore group, and that's of course because that's the most that's the only production and drilling we do in Norway is offshore. It's no onshore petroleum activities in Norway, only refining. So the upstream is only offshore. Okay, so this is some SIRs. For those of you who are not familiar with this, it's a effect measure that gives a understanding or gives a calculation of the excess risk. So we compare the incidence in the offshore worker group with that of the background population in Norway. So we can do this because we have the Cancer Registry as the background rate. So basically what this measure tells us is that is it more or less cancer in this occupational group than what would be expected for certain age, time period, and sex-specific rates in the whole Norwegian population. So if we focus on the bottom line, we see that there's a SIR of 1.7, which means that there's about a 7% increased risk overall among males. And likewise for females, it was about 13% increased overall risk. If you look at pleural mesothelioma, typically related to asbestos exposure, we see that there's a double risk, 2.38 in males. And in 2017, there were no female cases. This is a very, very rare cancer with long induction time. So it takes time to develop this kind of cancers. For melanoma, we saw that in female, there was about a 60% increase, SIR of 1.62 there. And for breast cancer, both males and females, we saw excesses, most prominently for males with a double increased risk. We have hypothesized that this, I mean, it's just 12 cases, but it is an extremely rare cancer form, male breast cancer. And we have one publication on this. I will touch that later, and it might be related to the extreme shift work patterns that we have offshore. In females, there was also a slightly increased risk, but that was not statistically significant. And then we see for the leukemias, the AML typically related to benzene. We found a quite remarked increased risk in females with an SIR of 3.76. We have also found increased risk with internal comparisons, but in males, this was not statistically significant. So we have here a 20% increased risk, but not statistically significant. For prostate, we found a 20% increased risk, and we published on that. And that we have found was quite certainly linked to rotating shift work, especially the duration of the rotating shift work, which is interesting and possibly could be explained by similar mechanisms as male breast cancer. So I will dive a little bit more into the etiology, the risk factor analysis, and the more etiologically directed analysis that we have done. So this is a paper which is now almost 10 years ago. It's from 2015, and I think the follow-up here was up until 2011. So now we have almost 10 more years of follow-up. At the time here, we only had 112 lymphohematopoietic cancers in the cohort, and now we probably have quite a few more. So this is what Besad will talk about later in his presentation, and what we're going to redo. So we want to redo this analysis, but also look at the interaction with shift work, because that was not done. We did not have the chronotox perspective back in 2015. So we want to redo this analysis with the shift work data employed as well. What we did find in 2015 was that the exposure levels were quite low, actually. So this figure here is showing that it's two y-axis. So on the left-hand y-axis, we have the GEM score, because this is a CMI quantitative assessment of benzene. So we had to translate this CMI quantitative assessment into average ppm. So you can see that the average ppm levels is quite low. It's below 0.05. We do know that for intensity and below 1 ppm for cumulative for ppm years. We do know that peak exposure is much higher than this offshore. So this is the average. It's important to consider. And we also do know that peak exposure might be related to acute hematotoxic effects. So it's important perspective to carry with us. On the right-hand side, we have the job exposure matrix that we have used for these calculations. And what we do is that we use the self-reported work histories, and we combine that with the GEM ratings that you see in the right-hand table, and then we arrive at what we call the GEM score in the figure on the left y-axis. So that's the data lying behind this figure that we see on the left-hand side. Okay, so what did we find in terms of cancer risk? This table is showing 3 subtypes of lymphohematopoietic cancers. So we have which has been known for many decades, the acute myeloid leukemia on the left-hand side. Although we had quite a few cases, just 10 cases, we saw a highly increased risk in the top tertile. So compared to the unexposed. The same also was seen for multiple myeloma to the right-hand side. It was increasing very much in the top tertile. And we also had a statistically significant trend across these tertiles on 0.024. We also saw an elevated risk pattern for chronic lymphatic leukemia, the CLL. However, this pattern was not that clear as AML and multiple myeloma. So that was the lymphohematopoietic cancers. We also done another study where we looked at aromatic hydrocarbons and risk of skin cancer. Here, the interesting finding was that we didn't find much on other sites of the body than the upper limb. So in the cancer registry in Norway, we registered these skin cancers with anatomic site, and we are limited to register them as the head or neck, the trunk, including the shoulders, the upper limb, or the lower limb. So we don't have registered or electronic, we have it registered, but not electronically available for more specific sites than the upper limb. So we went into these 24 cases at the upper limb, and we reviewed all the medical certificates, and we reclassified them into more fine resolution or high resolution anatomically sites. Specifically then the upper arm, the forearm, and the hand. And there was also one case that we could not classify in terms of anatomical subposition. The findings here are quite interesting because we didn't find any association between aromatic hydrocarbons for any other sites than the upper limb. So we interpret this that as it was local contact with the arms when you work and are in contact with the hydrocarbons, and then also local absorption of the hydrocarbons. That was the cancer initiation process. We also see that in terms of risk, we used a job exposure matrix for crude oil, and we also used one for benzene. Again, this is very few cases, as you saw here. It's based on only 16 melanomas and 8 squamous cell carcinomas, in total 24. And if you look at the upper limb, we see that for the forehand, arm and hand in the middle, for the upper arm, we didn't see much. So that is from the shoulder down to the elbow. That was not very much going on. But then we went down to the forearm and hand, and also the hand alone, and then we see that the risk pattern clearly changes. This is very wide confidence intervals and few cases, so the single estimates is not so important. Most important thing I think is the overall picture, and this strengthened the hypothesis that this is local absorption. It's basically when you touch the oil, the cancer process may be initiated. And although they use gloves and other protective equipment, it's very much often very soaked, so it's not helping that much. And also sometimes they take off the gloves because they want more precision to do the task and so on. Yes, and the next study I will present for you is about night shift work and chemical co-exposures. So here we are trying to incorporate the perspective of possible chronotoxicity between shift work and co-exposure. This is for breast cancer in females, so it's quite a small study. I think it was 80-something cases of female breast cancer, and it was low powered. So this should also be done again with a larger study sample to have more statistical power. We found some indications that it might be something going on in terms of shift work, little increased risk, but very imprecise estimate. So we conclude that we basically cannot say much about this. Here we have a table showing the breast cancer risk only according to the co-exposures. So we have the main occupational activity, and then we have chlorinated degreasers, which is basically tetrachloroethylene and trichloroethylene. But due to the few cases, we had to do a very basic analysis, just never ever exposed. And also we know that the assessment of the chlorinated degreasers was very coarse. So it's based.
2 minutes, please.
Okay.
Thank you.
I'll speed up. So I think I will just now, if it's only 2 minutes left, I will just jump a little bit ahead. I think we will skip this one. Yes, this is about the male breast cancer was doubled. I talked about that. This is about the prostate cancer. We see here that with the higher duration, above 19 years, the risk almost doubled. This is about more about the prostate cancer. We'll move a little bit ahead of that. This is new results from the bladder cancer study, newly accepted. we find a increased risk. This is not assessed in terms of shift work, but only benzene. This is quite interesting, I think. It's coming now in British Journal of Cancer this autumn. And we found an increased risk with increasing benzene exposure, both for duration, but also for cumulative, but not that prominent. This is for lung cancer. This is a manuscript that we're working on right now. We see an overall pattern of elevated risks, but not statistically significant, except for adenocarcinoma in relation to duration. So I'm sorry this was poorly planned in terms of timing, but just a few concluding remarks about the planned coming studies and the directions for the future. So we are now establishing a new cohort. So what I talked about today is only the NOPW cohort, but now we are establishing a cohort which is double the size. So we expect to have about 93 to 100,000 offshore workers who have worked offshore based on helicopter transports. So the nice thing about this new cohort is that it's register based. It's based on the heliport transport records, and in those heliport transport records we also have the shift plan. So we know whether they have worked night, day, or rotating shift for all of these 100,000. We struggle now a little bit to assess the exposure assessment for benzene because the job categories is not straightforward, but that's something we work on. And we also want to link these two cohorts to all the registers that you see in the slide, which makes assessment possibilities for many different outcomes, not only cancer, but also cardiovascular disease and the neurological disease and mental illness, and other respiratory diseases, for example. I think I'll stop there because I spent my time quite a lot, but thank you so much.
Thank you very much, Joe. Very interesting presentation. Indeed, this is a great work that has been going on for years, and it will continue going on for years. So congratulations on setting up this cohort and continuing on it. We will continue with the next presentation by myself. So allow me to share my screen. Hope you can see my presentation slide. So can you see it? Is it visible?
Yes.
Okay, okay.
Konstantinos, I think you shared the wrong screen. We see the your presenter mode.
Okay, let me see. Now?
We still see the presenter mode with notes and next slide and stuff like that.
I don't. Now?
Yes, perfect.
Okay, all right. So thank you very much for being here with me today. I will actually talk about some preliminary results as they come out from the Exposure Work project. This is a project co-financed by the European Regional Development Fund and the Republic of Cyprus. It's actually based on a proposal that received the Seal of Excellence in the MSCA called by the European Commission, and then was eventually funded by the Republic of Cyprus through the Research Innovation Foundation that allowed the postdoctoral fellowship of Dr. Behzad Haibadi to take place. And he actually did this work together with Samuel Abibola and Pantelis Harisiadis and myself. So allow me to start first by sharing some introductions. Let me see if that, okay. Provide you some synopsis of the work. I will start with some characteristics of night shift work. Secondly, I will continue with the relationship of these working habits with potential effects on some early stages of chronic disease, including inflammatory and oxidative stress processes, and how this, under certain circumstances, lead to tissue oxidative damage, coinciding or one leading to the chronic inflammatory response with downstream adverse consequences for human health, with some endpoints of disease already showing up in this graphic. So then I will continue with the chronomodulated metabolism of xenobiotics. In this case, the xenobiotics of interest will be the BTEX, and this is actually the main focus of the project, the exposure work project. And as I mentioned in the cover page, I will share with you some preliminary results of the exposures of BTEX in gas station workers in Bulgaria. And then we'll close with some results and future steps. Okay. So allow me to start by mentioning that shift work, according to the European directive, it's basically any method of organizing work in shifts where workers succeed one another at the same workstations based on the particular pattern, which is either rotating, continuous, discontinuous, and that allows the workers to work at different times over the given period of days or weeks. This working habit, as we have a figure here, shows how, let's say, a working habit that is not based on working at nights is maybe characterized by the regular homeostatic habits during the day, where you actually follow some day to night, light to dark, let's say, paths, and you adapt these light-dark habits to your behaviors, to your lifestyle. Whereas it is possible that under certain circumstances, disrupting this day to light continuum, such as in the case of night shift work, could also be implicated with changes in dietary habits, in sleep, obviously habits, drinking habits, and also some other habits that could potentially lead to a desynchronization of circadian clock with the peripheral clocks and the peripheral biological processes that take place, and could under certain circumstances lead to toxicity. This circadian disruption is actually a pretty common phenomenon if we consider that about 20% of the European workforce is actually engaged in some kind of a non-standard work schedule. And accumulating evidence on night shift work has been charged with circadian disruptive and also carcinogenic effects, as the IREC recently declared night shift work as a probable carcinogen. And this is based mostly on findings that appear to be pretty consistent in animal models, but also some significant results from human studies. So, talking about the association between night shift work and select biomarkers of oxidative stress, including tissue damage and inflammatory or metabolic response, has been recently studied in a scoping review we published a few days ago in Environment International, where we saw that indeed the majority of the selected studies on night shift work and inflammatory or oxidative damage health outcomes showed some association, but this association is not that robust, so we had some mixed results. And if we consider that oxidative stress and damage are part of early stage signals of a chronic disease process, then I believe we need to pay more attention to these temporal dynamics of human biology in response to these external stressors, such as the case of night shift work. But these habits, these working habits, these non-working habits are also co-occurring with other lifestyle and behavioral aspects of people's lifetime, which are equally important to consider, such as the rhythmicity of oxidative stress and damage processes that may occur because of these external stressor effects. And perhaps we can use a more holistic methodological framework, such as the human exposome concept, that tries to collectively comprehend and assess and characterize all environmental exposures and occupational exposures that occur throughout one's lifetime, including the endogenous response. And if we try to apply this human exposome paradigm to a night shift work health study, then we could do so by looking at the left, where the human exposome is typically composed of three major domains. You have the internal exposome domain, where one could classify in there circadian clock rhythmicity, relevant biomarkers, health status of the individual, the oxidative stress and inflammatory markers, and other internal, let's say, biochemistry or other components, systems biology, you name it. And this shall be coupled with a specific external exposome domain. In there, one can classify the details of the shift work activities, dietary aspects, sleep, occupation type and duties, other lifestyle and behavioral aspects. And in the general external exposome, one can actually include there the various policies of the industry, the regulations that are in place at the relevant work, climate issues, traffic, green space, etc. So having this in mind, I recently published an essay on how one can try to link the external exposome, let's say habits. These habits can take the form, as we said before, night shift work and other repeated during the 24-hour period habits and behaviors of individuals that may occur or not in critical life windows of susceptibility. And there is an interface here that connects these external exposome habits of individuals with the circadian clock, circadian oscillators, and metabolic regulators. And then this system of the circadian and metabolic function also interacts with downstream biological systems and processes that are part of the internal exposome, that if these processes, this continuum of processes and these combined interfaces are aligned and reach some kind of a chrono resilience, then no toxicity shall be observed. But when these interfaces are disrupted, you have some kind of a chrono disruption present. This, under certain circumstances, could be associated with a differentiated disease phenotype and could lead to a particular toxicity window. In searching these chronotoxicity windows, we have previously published some chronodisruptive metabolism studies on synobiotics. And to this extent, on the top right side, you see some results of a panel study we conducted on time-restricted cleaning activities on certain urinary biomarkers of exposure to disinfectants. So with gray color, you can see the bars that correspond to pre-cleaning activities of individuals. The horizontal bar within that box refers to the median value of that urinary biomarker of exposure. And the white box is after the cleaning has occurred. So when these processes, these cleaning activities take place in the morning, you see these unexpected results where the median values do not show the anticipated increase that you would see. And we saw this pre to post increase in the urinary biomarker levels when the cleaning activity took place in the dark at night. So we actually repeated these panel studies 3 times and we actually observed the same phenomenon. And then we conducted a more controlled study, a randomized control trial, this time with healthy adults, where we monitored the food contaminants in the fruits and vegetables that were consumed in specific times of the day. So this graph here, you can probably see my mouse, refers to the evening consumption of fruits and vegetables, 5 a day. These fruits and vegetables also contain these pesticide metabolites, the 6CN and the 3-PBA. With green is the baseline pesticide metabolite levels before that week's daily administration of the fruits and vegetables by the participants. But as you can see, when this fruit and vegetable consumption took place in the evening, 7 to 10 p.m., for example, you see what you would like to see, you would expect to see, the increase in the urinary pesticide levels compared to the baseline, but you don't see this phenomenon when you are administering the same individuals to consume the same fruits and vegetables during the morning from 9 till 12 noon. So these are some interesting results that led us to try to test this chronodisrupted metabolism in the case of BTEX. And these benzene exposures, these time-stamped BTEX external and internal exposures for night shift workers, we wanted to study their characteristics using the human exposome framework. And today I'm going to share with you only the results from the particular objective that refers to the influence of the night shift work on the magnitude and variance of BTEX external exposures for gas station workers in Bulgaria. So let me start by sharing some results from the methods. So this was a panel prospective study that we conducted in 71 workers of gas stations in Bulgaria and Plovdiv last spring. And we acquired bioethics approval and written informed consent for this study. This is a study design that we follow here. 2 days of day shift, 12-hour daily shift, then followed by 2 days rest, and then 2 days working at night. We also administered questionnaires to these workers on various lifestyle behavioral aspects. We used radielo diffusive samplers in order to capture the airborne BTEX in the personal air of the workers using this passive sampler that they were worn on the workers' lapel area. And at the end of the shift, 12-hour long, the cartridges were sealed and then taken to the lab to analyze them with the GCMS. Some results. These are the characteristics of the 71 workers. As you can see, most of them were males. Most of them were married at the time of the study. Their educational status for the majority of them was of elementary education. As you can see, the current smoking status was not high for them. And these were stationed in 3 gas stations in Plovdiv. And these are the airborne end-of-shift BTEX exposures in micrograms per cubic meter overall. These are the percentile data. You can probably focus your eyes on the median values for benzene, toluene, ethylbenzene. These are not above the regulatory safety limits at the workplace, as you can see. We had some pretty high maximum values, but these were not the rule. This was the exception. These are also the results of by shift work, whether the shift work was in the day or at night. We see here that the median values did not differ during day and night. This is actually a good result for us when we are going to analyze the urine samples of the individuals in order to assess that despite this external exposure, no difference between day and night, whether we can still report some chronodifferentiated metabolism of urinary BTEX values. And we also have results how they are differentiated by the current smoking status for all 5. So I'm wrapping up because the time is also pressing. We did not find differences in airborne BTEX levels by shift work, day versus night, in the personal air of gas workers. The magnitude of this airborne BTEX occupational exposure seems to be within the regulatory safety limits. Now, the next step is to actually test the hypothesis that the metabolism of BTEX values using the urinary biomarkers of exposure to BTEX would be unaffected by the 12-hour shift work schedule. And in addition to this, we are going to conduct some mixed effect models to associate the urinary BTEX values with oxidative stress and tissue damage biomarkers that we are going to analyze also in these samples. So obviously, I would like to thank the CII team, the funding from the Research and Innovation Foundation, and also to the local Bulgarian team that helped us a lot with the study logistics and sampling. Thank you very much for your attention. So allow me to proceed. We are going to have some time in the end to have a good discussion about all these presentations. So please allow me to continue with the next speaker in the agenda. And I have the great pleasure to have a presentation by Dr. Sana Rui, who is a group leader, and he is actually lead senior researcher at the National Institute of Occupational Health of Norway. Dear San, are you here with us? When you are ready, you can actually share your presentation.
Can you hear me or?
Yes, we can hear you, Sam.
Okay. So let's.
We cannot see you.
Yeah. So do you see my presentation or my screen?
Yep. Yes, if you click also on the presenting mode.
Okay.
Perfect.
And you can see it in presentation mode.
Yep.
Good. Okay, so thank you for giving me the opportunity to say something about, the biological mechanisms. I think I have just a bit changed, the title, which is changed it from cancers to carcinogenesis. And it is very ambitious, title, and I think it's very complicated as well to mechanisms to combine exposure to benzene and shift organ. Some of my talk would be based on general thoughts, and the others would be the data that we have generated here at our institute, and also try to speculate, some kind of common mechanisms for at least benzene and night shift work for future research to be considered. So I'm from National Institute of Occupational Health. Apparently, we are working with the occupational health, and we are dealing with the workers' health. So I hope this slide is, you can see it's, cancer is really a very complex disease. And as you have already, mentioned that many of environmental factors, such as, work environment, where you have chemicals, you have biological agents, as well as physical agents, may have effects, and then you have the internal factors, hormone stress, and so on, where you have the normal cell, which will, because of the effects of these agents, of course, possibly get a kind of genetic change, and then these genetic changes will go on, given that the exposure is present. And this is the belief for chemical carcinogens, at least. Most of the chemical carcinogens, they do cause cancer through a kind of multi-step carcinogenous process, which includes initiation, where you have small cells that still could be reversible, the changes there, you have mutations there, or they may go to pre-neoplastic cells in the promotion phase and may lead to carcinoma in situ, and eventually, if the exposure is still there, so it may go to a metastatic phase. What is important is here the genetic factors. When you need really genetic alterations in all of these stages. And from the literature, from the data, from the research, we know that there are many genes, important genes like tumor suppressor genes, DNA repair genes, oncogenes, which are actually on the type of proto-oncogenes, different ones, and then they will be activated even by the exposures. And also you have the other genes that may lead, to metastasis. So this is a very complicated process. Most of the the International Agency for Research on Cancer, IARC, they do a classification work, and they have, up to now, I think they have classified more than 500 agents as carcinogenic, meaning that confirmed, group 1, or probably group 2A, or possibly group 2B. What is evident when they reviewed the mechanistic data from all of these, not all of this, but some of these carcinogens, they found out that most of them, they have 10 key characteristics that are present in these agents. And this is very important, chapter in the work of IARC for classifying the carcinogens, because the mechanistic data are getting a kind of weight in their classification, in addition to epidemiological animal data. So that's an important work. So what are these key characteristics? These are given here in this figure. Most of these carcinogens, at least some chemical carcinogens like benzene, which is mentioned and was talked about, they are electrophilic and they need metabolic activation. And then they are genotoxic because they damage DNA. They also affect the DNA repair process and lead to genomic instability, which is a hallmark in many of these lymphatic, cancers, including leukemia. And the other thing is, the fourth key characteristic is that they induce epigenetic alterations in DNA and also in the proteins, methylations in DNA and histone modifications in the histones. And one important mechanism which was mentioned by Konstantinos is the reactive oxygen species, not only reactive oxygen species, but of course, reactive nitrogen species. So totally, we are talking about the induction of oxidative stress and also induction of chronic inflammation. So one thing we should remember here is that the acute, for cancer to arise, so the acute inflammation should lead to a chronic inflammation, a chronic inflammatory status. And also the seventh key characteristic of these carcinogens is that they are immunosuppressive. Of course, some of these agents, they act through the receptors, they affect the receptors and causes immortalization. This causes immortalization. This is one of these one of the properties of the cells, as I was mentioning. When the cells become neoplastic, so they really become immortal. So this process, we have been looking at the night shift workers, working nurses. And the 10th is that these carcinogens, they alter cell proliferation, they affect cell death, for example, apoptosis and so on. And also they affect the nutrient supply that the tumor cells need for their growth. So not all of the carcinogens actually are having all of this. Some of them may have only one of these key characteristics, like TCDD, dioxins, whereas other agents like benzene, which I will show you another slide later on, may have many, many more key characteristics of the, like benzene will have 8 key characteristics. The same, I think, is valid for asbestos, maybe not affecting not 8 key characteristics, but at least many more than 1. So what about the benzene? I mean, you showed epidemiological data, but again, as I was showing in the first slide, so benzene is also kind of a complex chemical. It is metabolized in liver and lung, and of course also in bone marrow. It is metabolized by cytochrome P450 system. And of course, most of these metabolites are detoxificated by different enzyme, GST, and microsomal epoxy hydrolysis, and so on. But if the metabolites are not detoxificated or removed, they may lead to reactive compounds like we have it, benzoquinones, and they may lead to toxicity. When we are talking about toxicity of benzene, here I particularly talk about the different cancer types that they may lead to, and you was showing nicely some data from their results that so many cancers could be related to exposure to benzenes, at least from the epidemiological data. So the benzene has a multiple mode of action. As I said, the key event which happens is the exposure to the benzene and the metabolism. So when we are talking about the pathway analysis, especially in leukomegenesis induced by benzene, so we are talking about the metabolites of benzene, which may lead to many other key events, like dysregulation of stem cells, genetic instability, and so on. And then we will have, the cancer here at the end. So here in this slide, I just highlight the importance of reactive oxygen species. Again, I mentioned nitrogen reactive species as well, they are important. And you have genotoxic stress and epigenetic changes that are very important in the process of initiation and also progression of these leukomic cancers. So now get back to the night shift work. Night shift or shift work was classified as a group 2A in 2007. However, IARC again convened in 2019, and they kept the same classification because there is limited evidence in humans, but sufficient evidence in experimental animals, as well as, even though it's not mentioned here, but the mechanistic data were also supporting this classification of group 2A, which is probably carcinogenic to humans. The same in this meeting 2019, I think there was also found positive associations between prostate cancer, colon, rectum. One thing I should mention here is that in this 2019 meeting, it was specifically talked about night shift work. In the previous 2007, it was shift work that is associated with disruption of circadian rhythm. But here it was specified that night shift work is probably carcinogenic. And we know that night shift work, as others have taken, it is in many workers are affected, nurses, factory workers, flight attendants. There are some data, airplane pilots, and so on. And of course, in many sectors in the industry, manufacturing and transport and so on. So we are kind of, 24-hour society. And again, like benzene, shift work or night work is really complex. And the complexity is also even because you have other not very classical factors, but you have this, psychosocial factors there, sleep disruption, you have altered meal timing, and also other social behaviors. So this is also complex as benzene. So what we have done at STAMI, our first study was to look at we had a Norwegian nurses cohort, and we wanted to analyze this data, the risk of, or at least the risk of breast cancer in night shift workers, in nurses who work in night shift, with another exposure metrics. Here, what we did was that we looked at the number of consecutive nights. So what we found was that the nurses who had worked more than 6 or more than nights consecutive and for more than 5 years, so they had really almost twofold higher risk of getting breast cancer.
Dear Son, 2 minutes, please.
Okay. So this is then we looked what could be the mechanisms of this risk of increased risk of breast cancer. So one of the things was that we just hypothesized that this circadian disruption due to the night shift work may be important. We looked at polymorphism, and we found several positive associations with genetic variations in circadian rhythm pathways and the breast cancer risk in nurses. And then we looked at DNA methylation, where we really looked at many genes in the circadian disruption, and we found several positive associations where we found methylation of, for example, very core circadian genes like clock, BMAL, and CRY1, which is very central in regulating the circadian system. And then when we looked at data, looked at that, so we found the kind of plausibility that this disruption of this clock system or circadian pathway system, it may lead to disrupted DNA repair, cell cycle, and also cell proliferation and differentiation. So there is a biological plausibility. The other thing that we looked at was telomeres. Telomeres are repetitive sequences which are protecting our chromosomes, and by exposures, they get shorter, and as well as by the age. So what we found was that, I will just skip this and go to our own data, was that if we looked at the number of consecutive nights that the nurses had worked in relation to telomere length, then we saw that shortening of telomere lengths related to the number of nights that nurses had worked. And this shortening, we could see it only in the cases, in breast cancer cases, not in the controls. So what are we know that, night shift work is really very common in many industrial sectors where exposure to other chemicals like benzene is there as well. As I'm mentioning, so benzene is affecting at least 8 key characteristics as indicated by IR. And the night shift work, when we look at night shift work, so the similar or sometimes the same key characteristics are also affected by night shift work. And this is based on the data from animal studies and also epidemiological data. So my hypothesis is that there is a kind of possible common network pathway between exposure to chemicals. Here I have taken the example benzene and night shift work. Even though benzene may directly affect these key characteristics, but night shift work from a kind of indirect way by affecting the circadian disruption pathway. I think this is the main my conclusion, that this is very interesting pathway mechanistically to see what is happening in the night shift workers that are exposed to benzene or other chemicals in offshore and other sectors. So thank you for that. This is our group, Occupational Toxicology at STAMI. And thank you very much. This is sunset from Oslo from top of a hill.
Thank you very much, Dr. Naroui. Very interesting presentation. Indeed, there are some plausible biological mechanisms of cancer that maybe seem to be common for night shift work and perhaps BTEX, so it's worth exploring further. I would like to continue with the next presentation. As I said, we're going to leave some room for discussion. I already have several questions for the speakers, but let's continue with the next excellent speaker. We have Dr. Ioannidis. He's the managing director of the AEI Scientific Cultural Services here in Cyprus, and he has a very unique and related presentation to share with us today. Dr. Ioannidis, you are muted. Can you unmute yourself, Dr. Ioannidis? So I'll be on the lower left side, maybe.
Okay, first of all, Konstantinos, thank you for the invitation. Let me just try to share the screen while I have the slides on. Because I had some difficulty with that a minute ago. Okay, so. Can you see my screen right now?
Yes, it's coming up. You see only contact details.
You have to go at the beginning. Yeah. Apologies, I was trying to do some changes at the last minute on the slides here. Okay, thank you for the invitation. It's really wonderful to be here and to have the opportunity to share this podium with these excellent speakers, and in a topic which, thanks to the work that we've done recently with my colleague Ligia, that is going to talk after me, it has extreme interest to us because we can see some connections with some work we do about the brain with the work that you are doing. I will let Ligia describe most of the things about the project that we did, and I will try to bring together some of the results we had from a number of works recently about sleep and about how sleep affects behavior in general and the health of individuals, in order to bring some similarities with what we heard before with the excellent things we have, the excellent talks we had so far. So I will start with the main message because, in the interest of time, I want to pass the main message first and make sure that this is heard. When we have an environmental threat, you have two distinct things that we need to do in order to minimize the consequences. First of all, you have to recognize what is the thing that what is the threat, and then what is the fraction of the population is likely to be affected, epidemiology. And that seems to dominate everything. But there is another part which I want to emphasize, and that is the immediate response for the people affected, which has to be personalized. Okay, it has to be treated as individuals, but very often this is forgotten because in an emergency this is not always easy to do. So my message is for the second category, which must be put on case-by-case basis and where personalized approach is necessary. Thanks to relatively recent advances in non-invasive electrophysiology, particularly in the tomographic analysis of EEG, very recently some work from our laboratory, and directly from magnetoencephalography. There are some exciting opportunities to actually do that are already available. For the purpose of today's meeting, the role of sleep in this approach would be emphasized. So first of all, let me tell you something briefly about the techniques we use. We can measure the magnetic field generated by the brain using very expensive equipment. You can see here a schematic of magnetoencephalography. You have a helmet-like instrument which records the magnetic field around the head. Everything is embedded in liquid helium. You have to, the signal you detect is minute, but it comes instantaneously to the sensors. So you can do millisecond by millisecond tomography, provided you can eliminate the much larger environmental noise. We can do that. So it's not time to describe the details, but please believe me, we have a technique of doing tomography with imaging data, which we published for a number of years. It was very computationally intensive, but nowadays this is not the problem. The problem is how to deal with the image. At the end of the day, you can end up with a real-time image of what's happening with the brain, millisecond by millisecond, which is in the right order in order to see how information is transferred between areas of the brain, and in millimeter accuracy. What that means, it means that we can see the geographic countries of the brain, where the cytotonic areas which define the processes, activities, and how this information is transferred from one such area to another. So you can see the static thing where things are activated, how communication happens. It's still very expensive. It's not very easy to actually have this huge number of images and make sense out of it. Maybe some AI techniques will come later. But what we emphasize so far is that the tomography, this is an example of when we demonstrated the agreement with fMRI in the order of a few millimeters. Again, it's the references are there. The next one, using that, we can now go and find something new. For example, in this presentation, we have demonstrated that if we look at the activity as we've seen in the previous slides, the fMRI, which that we can identify exactly when and where the activity is coming first in the cortex, namely at about 40 milliseconds. But what is most important is when we've done exactly the same analysis, real-time analysis, and look at the comparing when the subject is paying attention to the side of the stimulus, on the left, for example, here, compared to what's happening if he pays attention to the other side, we find exactly the same activation at exactly the same time, showing that the first activation into the brain is affected by attention, which is totally turn around what people believe up to that time. And the reason is because we looked at single trial real-time activity, while other people use at average data where all this detail was lost. We can look at activity everywhere in the brain, and finally, in terms of just show what the technique can do. Here is a result about the generators of the K complex, a big event in the brain. This is on the left is the result of a huge study, 15-year study of epileptic patients in which we have activation in the brain, and the only areas identified is what we can see here, left, and just magnified a little bit to see it more clearly on where the arrow is. By only four subjects, with 49 millisecond by millisecond imaging of the brain, we can identify almost exactly the same area as far as I can see from these diagrams. And here you can see some zoom versions of those. That's quite remarkable in itself. But okay, let's now go straight into sleep. When we started studying sleep for the first time in this way, nothing could make sense. We looked at the large graph elements, the large events in the EEG and the MEG, and we couldn't really see chaos. We could localize, we knew that, but we could not really put all these activations in order, except when we started looking not at the large events, which are basically the chaotic events which happen when the brain is in a stable position as it goes back to equilibrium, but when you look at the quiet period. But to cut a long story short, the first thing and the only thing that we see which makes sense is this progression of gamma band. Gamma band activity is high frequency around 40 Hz activity, which tends to occur when you have cognition, which seems to increase from sleep, increase, not decrease, when you go to sleep, starting from very low values, and then increasing progressively, becoming huge when during REM sleep. The importance of that investigating furthermore is that this seems to indicate that these two areas there, one at the front of the brain here and one at the back, seem to be the areas where the core of the representation of our experiences is kept. And this, to my mind, sounds like the new representation of self for reasons which I can answer questions, but not right now. It is not the default mode network. The default mode network is all around it. When we put the default mode networks that people talk about right now they can see the theory of mind areas yellow around it and beyond the lower default mode networks in both cases. This seems to be at the center in an area where the default mode network has no representation. Actually, it's the other way around. The way we interpret it is the more you go outside these two centers, the more away you move from the new representation of self, you come to areas which are basically the slaves of self, which carry out what the self wants to do while the self is asleep when we are awake. I'll come to that in a minute. So the idea so far, we must sleep safely to make the events of the previous day parts of ourselves. That's memory consolidation. But one of the most important things about sleep is that we have to keep safe. So we have to keep away from danger while we are immobile. But at the same time, we have to do something very important. The analysis of this seems to indicate that everything is taken care of in order to dispose of the influences from the environment for something very important to happen during the same stage that the K complex are occurring, and that is spindles which are related to the memory consolidation. The frontal area is sharpening the context to be stored, where the posterior areas of the brain sharpening the memories to be stored for later recovery. Memory consolidation during sleep is for the integration of salient events, not just the ordinary events, which happen for the internal image of the work and the new representation of self that must be minimally changed in the process. The consequences, the new representation must be from early life. We have evidence for that again. I have some slides, but no time to spare them. The self must be protected from the vagaries of the awake state because its co-representation must be preserved. The failure to do so lead to pathology. I know I'm covering things very quickly, but the essence of the message is that while we sleep, our self is reconstituted in order to make the changes that are necessary because of traumatic experience during the day. Then it goes to sleep when we're awake, and the slave carry out whatever the reconstructed self has set up to do. So the identity of integrity must be protected. The new representation of self must be handled with extreme caution and precision. Memory consolidations only for salient events must be done at special, safe times during sleep when the external environment is minimized and there's no other interference, because if it does, then the salient events would become confused with other things, and then mood pathologies and psychiatric doctors will occur. Minimal necessary changes are therefore made. The changes are integrated before we wake up. That's why in REM we have all this huge increase in activity. It's where the new self plates itself scenarios. That's why usually in dreams are rather extravagant, because it has to prepare for the next day, for possible events of the next day. The new representation is dormant while you are awake, and it wakens up during the next day. Predictions. Here it comes to what might be of interest to this community. Severe or persistent negative experiences make the reconstitution of the new representation of self difficult. That can lead to mood disorders. Changes in the environment are also things which the brain recognizes as very dangerous. So they fall into the same category. And changes in the environment could be medication. That's why you have, for example, others, and also the things that we are studying in this webinar. So new chemical substances in the environment are things which don't fit the general pattern. There is nothing in the new representation of self that can accommodate that. So in response, agreeing with the previous speaker, I would say that this mismatch between the new representation of self that has to be modified according to dramatic events and the instance of the chemical insert, that's where all the changes that we described so eloquently in the previous lectures is likely to be occurring, and there is a lot of mileage to be done from those two. So for medication, we have the evidence that this is happening. For this one, I hope we can produce some evidence working together. So we have two ways of doing the analysis. We can go into the source space, a very expensive way, do the tomography millisecond by millisecond, and that's what we usually do. Or we can work on signal space. And this is what I would like to discuss very briefly with you. I don't want to take time from my next speaker, which is my colleague Ligia, who is going to talk about the project that we did. But because of various reasons, we only did the signal analysis at this level. We would like to do it, and for that we need a lot more subjects in order to have something worth doing. So for this work that we've done with Meftiadis and leaders here, And very quickly, I will skip the introduction because it will be covered in the next talk and method. In terms of electroencephalography, we use the genome system with 32 channels. We collect the EEG activity in resting state. I will describe that in a minute. Electrophysiological analysis, we substitute, we have questionnaires and we relate to resting state in what you see in a minute in only one small part of the experiment. Now, I have to remind you that the experiment was done during the COVID, and we had to not just wait for the events to happen, because we have to prepare, make arrangements with petrol stations. Constantino, you know that in Bulgaria, it's not an easy thing to do. So we have to anticipate weeks in advance when there would be a break in the lockdown in order to go out and do the experiment, not actually plan it and look at the neighbors around, but at least organize with the petrol stations to have enough, some good number of subjects. That's what we did. The experiment we did, we had eyes open, closed, fixate, resting state period, which was the control, then a number of tasks, and then repeat that ahead. What I will show is just the results of this first analysis at the signal level only. So the analysis with flashy and ambiguous only at this early part. We have the data to analyze further, but we need more subjects for the huge effort to be done. And we're going to look at only two parts. In this one, you see the activity for just the first two periods of 30 seconds. 30 seconds of eyes closed, 30 seconds of eye open. We get the mean value for this, and we have the black line here, which shows the kind of threshold. And we look at what happens above this line. So we have how much power we have in this part, and then what is the duration of these excursions above the 2 standard deviation of the eyes open condition. This is one of the parameters we use. The other is the reactivity. Is how does the change occur? What is the change in one frequency stage from between eyes open and eyes closed? That's the reactivity. So we've done a PC analysis. Basically, we put all these parameters into a big box, and then we wait to see what this will produce. And here is going to do the rest. I will just show 2 results. First of all, the mean duration, as I described earlier, how that classifies it. Then you can see that the people who were on the pumps, the open circles, differentiate quite nicely from the rest. And if we do the same with the reactivity, we also have similar Ligia, I think, is going to show a curve where he describes exactly the same results, but now compared with the chemical analysis that she did. Okay, this puts the two together. And as an end to this, I'm hoping that by this coming to this very rapid exposure to what we do now, what we could do in terms of analysis, there would be enough mileage to some European collaboration to actually put this kind of EEG analysis into some of the work that you're doing. And I thank you for your attention, and sorry for carrying so much, but I really wanted to keep in time and leave time for questions. Thank you.
Thank you very much, Dr. Ioannidis. No, you were right on the point with the time and with the content. Thank you very much for that. And let's continue with our next speaker. Is Dr. Ligia Eleftheriou. She's a senior analytical chemist at the CP Food Lab company Ltd here in Cyprus. And she will share, as Dr. Ioannidis mentioned, the chemical, let's say, exposure results for the BTEC study you did here in Cyprus. The floor is yours.
Thank you very much.
Dr.
I think Dr. Ioannidis needs to stop sharing his screen, otherwise I can't share mine.
Immediately. Constantino.
Ναι. Down.
Sorry, it's another Constantinos here for help. It's a kind for help.
Okay.
Where is it? I'm going to stop sharing. Okay, here we go.
Okay, Ligia, I think you can proceed.
Thank you very
Thank you.
much. Can you see my screen now?
Yes.
Yeah, thank you very much. First of all, thank you very much for the invitation and the very interesting presentations we saw so far. I'm Ligia. I work at CP Food Lab Limited in Cyprus, and I'm going to be presenting some results for the Plan CMB project that we did some time ago with Professor Ioannidis. It was an investigation on the effects of volatile organics and specifically BTX on the human body. It was a project that is co-funded by EU and Cyprus through the Research and Innovation Foundation. I'll give a short presentation on the participants, an introduction to the project, some sampling and development of the analytical methods, and then the results. So it was 2 participants, CP Food Lab Limited and AAI Scientific Cultural Services, as Professor Ioannidis already mentioned. AAI is doing basic and applied research in cognitive neuroscience, while we do food, environmental, and pharma analysis here in Cyprus. So the lab is a private laboratory. It's the biggest in Cyprus. It's accredited under ISO 17025. We operate at our own premises in Nicosia. We've got a range of instrumentation, including ICPs, LCMSMS, GCMSMS, various LCs, GCs, many instruments, as you can imagine. And then in addition to routine analysis that we perform for samples of food, pharmaceuticals, and environmental, we also participate in research projects, EU or Cypriot funded ones. Plan CMB was one of the latest ones that we participated and we led, and it's been awarded by the RIF. Also, as Professor Ioannidis already mentioned, AAI Scientific Cultural Services is a research-oriented private enterprise in Nicosia. They've got 2 big labs, the Laboratory for Human Brain Dynamics and the Cyprus Neurofeedback Center. But he's already mentioned all about the company. So I'll move on to the actual project. The Plan CMB, actually pollution levels at near and near gas stations and their effects on the human mind and body, was an attempt by us to examine how the emissions of gas stations are affecting the workers and also people living in close proximity to the stations. So we were looking into BTX, VOCs, MTBE. We did an environmental survey, and also we did blood analysis, questionnaires, EEG analysis to examine the EEG, the brain activity, sorry, and the behavioral mood of the participants. The scope of the project was to measure the BTX VOCs at the actual gas stations that participated in the project and also near and in the surrounding areas, to investigate the total exposure of the gas station workers to the contaminants, to explore the correlation between the pollutants and the EEG measurements that we did, and then also to estimate how the surrounding areas and the population living close to the stations is affected by the emissions of the station. Obviously, Professor Ioannidis already mentioned some results for the EEG measurements and how they related to the rest of the biological and environmental samples. I'm going to focus on the other side, so the analytical work that we did here at the lab. And then here is an overview. This is a map of Nicosia, and you can see the different locations that we targeted. The purple dots are the stations that participated, and the pink ones are the people living at the surrounding areas and in different distances from the selected stations, so that we could have background measurements and also measurements in specific distances from the selected petrol stations. We did focus environmental monitoring, as in selected locations within the petrol stations that participated and non-focused ones. So, and people living in selected distances from the stations. We did blood analysis for all the participants, workers, and the general population, and also mood and EEG recordings for all the participants. Now, the selection criteria for the petrol stations, we have several selection criteria, as you can understand, but I have to mention here that a very strong criterion was the willingness of the owners to participate, because we had a lot of difficulties for people to allow us to enter the petrol stations. And then here is a sampling, the plan that we did, and the three selection one, the petrol stations, you can see with access on the three maps, the locations that we chose to sample at from the individual petrol stations. As you can see, we have points in the center of the petrol stations. We have points around the petrol stations. I don't know if you can see my pointer, but as you can see here, we've got points where we sampled at around the stations, at a distance from the stations, and then further away. So the three stations that participated, you can see the individual locations. And then, okay, obviously we did some method development before we started the analysis for all the VOCs, BTX, all the analytes that we wanted to test. I won't go into detail, but I'll just mention that we did active sampling, and then we analyzed the air samples from the active sampling through GC FID. We also had instant sensors for VOCs and other contaminants, where we sampled on the spot the air quality. And then we also did some GC-MS analysis through perch and trap for blood samples for the ones we collected from the participants. And the EEG that Professor Ioannidis already mentioned. Now here you can see some images from the sampling that we did for the environmental monitoring. You can see in the blue circles our instrument, which was actively sampling air for 8 hours according to the European regulation on air quality. And then we also did the biological samples. We had some nurses who were withdrawing blood from the participants. And then again, the EEG measurements that were performed at Professor Ioannidis' premises or at the actual stations, depending on the people's choice and what the timings that we had. And I'll go straight to the results. So we had some trends. We have some trends that we expected, including how the distribution of pollutants related to the petrol decreased as we moved away from the center of the station. We also saw higher concentrations of pollutants during the supply to the cars, which was expected, but we hoped we wouldn't see that because companies nowadays follow specific regulations and techniques to avoid these emissions of pollutants when the cars are supplied. I heard Dr. McGree's earlier saying that they didn't see any difference between the night and then day samplings when they did the similar experiment in Bulgaria, but in our case, we saw lower concentrations during the night. And obviously, then we did some statistics to see what those concentrations meant, 97% of the samples that we analyzed from the petrol station showed very high concentrations of VOCs, which were much higher than the directive, the EU, what the EU regulation stated, and then much lower percentages were low concentrations or moderate concentrations, as you can see on the table there. And then I've got this link here, even though we did some mapping of the data, so you can see how through Tableau, how the distribution of the selected petrol stations was changing if we move from the center to the surrounding areas, which is what I already mentioned about higher concentrations in the center. You can scroll through, we can do that, but I think the time is not permitting that, but I could show that if you like later on. So I'll move to the next slide. And then in terms of the biological samples, we had, this is just the percentage of the participants. So 43% of the participants in this specific project were petrol station workers. And then we have, you can see there the distribution of the participants, 37% of the participants were not petrol station workers, but lived very close to the petrol stations, less than 1 kilometer away, 7% less than 3 kilometers away, and so on. And then these are some of the results. These are the results specific for the petrol station workers, where you can see with yellow there that for all the BTX that I'm showing here for benzene, toluene, ethyl benzene, and the xylene, that we had very high concentrations of the BTX found in the participants' blood, and these are specific to their workers, compared to much lower concentrations that we found in the people living far away from stations. Now, I have to mention here, this is not the case for smokers, so we could see similar results to the workers with smokers, and we had to do this distinction between the petrol station workers and the, say, control group. Here it's a summary of what we could find, like the PPMBs of the different BTX found in the blood. And then this is also what Professor Ioannidis mentioned earlier. So for the EEG measurements that Professor Ioannidis' group performed, we could see direct relationships between the, obviously you can say more about this, but we could see indirect relationships between the VOCs found in the environmental monitoring and the brain activity. And then we did some more focused PCA analysis to see how we could relate the blood findings with the environmental measurements. And specifically for xylene, for example, we could see a very good correlation between xylene and the VOCs in the environment. So a sort of summary of all this, we found that BTX seems to be the ones more persistent in human blood. There is a relationship between the EEG measurements and the brain activity related with the environmental VOCs. We had some trends relating the petrol station emissions, higher concentrations during day compared to night, lower concentrations as we moved away from the center, high release of VOCs during car supply compared to no car supply. And then we had obviously higher concentrations of BTX in blood of the petrol station workers compared to the control subjects, the people that lived away from petrol stations. Thank you very much.
Thank you, Dr. Eleftheriou. Very interesting and novel for Cyprus, but also I think for other countries, because you nicely considered both external and internal BTEX exposures for workers and also nearby resident groups. And you nicely try to associate those with brain activity, with Dr. Ioannidis' expertise. Obviously, this is a very, very nice project, and I commend you for that. So allow me to call on the floor the last but not least speaker of our webinar before we start the discussion and the questions, Dr. Behzad Haibati. He is currently a postdoctoral researcher at the Section for Epidemiology and Prevention at the Department of Research within the Cancer Registry of Cyprus, and he's going to present to us their analytical strategy to conduct a new study on co-occurring exposures to benzene during night shift work and the risk of a suite of lymphohematopoietic cancers in their Norway offshore petroleum workers. Dr. Haibati?
Can you hear me?
Yes, yes, we can see
The
it.
presentation is okay.
Yep.
Okay. Hello, my name is Behzad Haibati. I am postdoctoral researcher at Cancer Registry of Norway. The title of my presentation is Analytic Strategy for Study of Benzene Exposure, Night Shift Work and Risk of Lymphohematopoietic Cancer in Norwegian Offshore Petroleum Workers. In recent years, there has been increasing evidence of cancer risk associated with exposure to hydrocarbons such as benzene. As you know, benzene is volatile and colorless liquid, naturally found in crude oil, gas, and condensate, including produced water during offshore drilling and production. According to most recent review with the WHO, IARC, benzene is causing acute leukemia in humans and suspected cause of other histological subtype of LH cancers. And also there is a question about are workers with nighttime schedule particularly vulnerable to benzene toxicity due to the circadian rhythm disruptions. There is a study from Cancer Registry about association between night shift work, male breast cancer and aggressive prostate cancer. The study found that prostate cancer, especially associated with longer duration of rotating shift work schedule. Also, IARC working group has concluded that night shift work is probably carcinogen to humans. There are several studies from Cancer Registry of Norway about the association between benzene exposure and various cancer among offshore workers, which employ between 1965 and 1998 using Cancer Registry offshore cohort established in 1998. But there is a lack of studies on exposure-related analysis of LH cancer with the work history after 1998. It is also unknown how shift work may alter the benzene LH association. In addition, there has been general reduction in chemical exposure after 1998. So time-dependent modeling of benzene LH cancer association is important. We will all address this limitation by enabling cancer risk assessment related to exposure after 1998. So the overall aim of this project is to conduct a study of benzene night shift work and LH cancer. We hypothesize that there are those risk-response relationship between increasing night shift work, benzene exposure, and LH, and increased risk of LH cancer in offshore petroleum workers. In addition, we hypothesize that shift work influenced the association between benzene exposure and risk of LH cancer in offshore petroleum workers. The specific question that we have in this project, we have three specific questions. The first one, is there a dose-response relationship between increasing night shift work and risk of LH cancer in offshore petroleum workers? Is there a dose-response relationship between increasing benzene exposure and LH cancer in offshore petroleum workers? And the last one is, does shift work influence the association between exposure to benzene and LH cancer in offshore petroleum workers. Study population and data collection. We will utilize two cohorts. Norwegian offshore petroleum workers consisting of 25,500 men and 2,500 women who have worked between 1965 and 1998. New cohort, which is named Heliport. We will use another cohort, including all Norwegian including all Norwegian petroleum workers, which is about 80,000 individuals from the period of 2000 to 2021. The new cohort, which is named Heliport, will be based on the transport record from onshore heliport to offshore petrol and back. A recent linkage to cancer registry main database also revealed that between 1953 and 2021, around 9,300 cancer cases in total happened in combined cohort with around 90,000 workers. All information related to cancer diagnosis, data and cause of death, and data of immigration are available in cancer registry, the cause of death registry, and Norwegian population registry for the past 60, 70 years. We will all link these cohorts to this data registry to obtain information related to cancer, death, immigration, and so on. We will categorize each worker per you consider standard work lasting 14 days with 12-hour shifts. We will categorize either as day shift work, they are working 14 days of day work starting from 7 a.m. to 7 p.m. and night shift work. They are working 14 days night shift, which is starting 7 p.m. To 7 a.m. and rotating shift for they work 7 days of day work and 7 days of night shift work or vice versa. We have also mixed work classification. For the exposure assessment, we in brief we use a semi-quantitative assessment. The estimates that the occupational group has been developed for the period of 1970 to 2000. After 2000, an updated job exposure matrix will be developed. Then the scores will be translated to the ppm in order to compare with the previous studies. And work histories also will be obtained from the questionnaire from Norwegian Petroleum Workers Board. And for the heliport course, we will use helicopter transport records for work histories. Other information such as work outside offshore and smoking and other also will be derived from questionnaires. We will have 7 classification for exposure. The cumulative exposure, cumulative peak exposure, exposure duration, and average exposure intensity, and average frequency score of peak exposure. We will classify LH based on most recent WH classification of hematopoietic and lymphoid tissue by using histology code from ICD Oncology. Study design and statistics. We will use a cohort and case cohort study design to compare the incidence of LH cancer according to exposure categories of exposure to benzene and shift work. We will use CUT-CII regression adapted to stratify case cohort design in order to estimate hazard ratios associated with benzene metrics and shift work variables. In all our analyses, hazard ratios will be adjusted for potential confounding factors, such as age, benzene exposure from other works. We will also use DAG framework in the modeling before our analysis to identify potential confounding factors that should be adjusted for. This is our article, which was published a few days ago in Scoping Review on Night Shift, which was published in Environment International. We find that night shift work is associated with biomarkers of oxidative stress, damage, and inflammation. But as Konstantinos mentioned, there are mixed results. There are several studies on oxidative stress damage and night shift, but still there is a need for comprehensive investigation into the temporal aspect of oxidative stress damage process triggered with environmental occupational stressor in human studies. That can be also included in the future by using Norwegian data. So in fact, biological monitoring for exposure is advantage because it permits estimation of dose and burden from exposure throughout all relevant portals of entry. So this can be also investigated in the future by using Norwegian petroleum worker data. And we found also in our scoping review that work-related exposure are overlooked in exposome studies, despite that working life cover most of the life expectancy spans, where work exposure can be high in magnitude and frequency. So it is important also to include the exposome framework in the future in cohort studies and implementing current toxicity aspects. Also, we suspect effect modification, so it will be interesting to estimate the interaction between two or more chemicals, for example, benzene exposure and night shift work. There's a study study on flexible meta-regression in order to assess the shape of the benzene leukemia exposure response curve. They find that there's they stratified analysis, and they find that they indicated a considerable impact on the shape of the benzene leukemia exposure response curve. But it is unknown how this stratification by shift work, if there is an impact on the shape of the benzene exposure, benzene leukemia ERC also, that can be also included in the future by using our data. I think it was my last slide. Thank you for your attention.
Thank you, Dr. Haibati, also for the quite ambitious analysis plan for within the cohort, the Norway cohort. So now it's the time that we all have been waiting for. Finish with the presentations and now open the floor for questions and discussion. So anyone who wants to address a question to any of our speakers, feel free to do so. Who would like to go first to ask a question or make a comment?
Can I just have maybe I may start,
Please.
You and Mesat, I think it's very nice, studies, I mean, you have done already, and they are very much in line with what IARC has been trying, to do. I mean, kind of, somehow to give weight, to the kind of, mechanistic data in their classification. But one thing we should remember regarding the IARC classification, IARC is doing the hazard classification. It's not the risk classification. I just wanted, to mention this, I mean, to the entire, the entire, attendees here, because the risk classification is done, by the companies or by the enterprises. They are there. So IARC is doing only the hazard kind of, classification. So this should not be exchanged. And the other thing is that the Nordic expert group for criteria documentation of health risk from chemicals, which is increasingly becoming more important in relation to this, occupational exposure limit values. We know that the occupational exposure limit values for benzene, they have been really dramatically, decreased. And that's very much in line with what you have, you're trying to, investigate whether these lower exposures, because in Norway, at least, I mean, the exposures are really strictly, regulated by the authorities. And the exposures, whether this dramatically lowered exposures of workers to the benzene and possibly other products like, Vitex and so on, whether they reduce, they lead to kind of, reduction in the risk estimates. That's very, very important thing. So me and Magne Brodtvedt, your co-authors, him and one of my colleagues from STAMI, we were asked to look at the occupational chemical exposures in combination with unusual working hours. It was an assignment from the Nordic expert group. And the idea was to look to see whether this occupational exposure limit values should be different, where there is a combined exposure to, let's say, this hydrocarbons and night work. And when we reviewed, there was really there was no specific literature which had handled this topic, a combined exposure. And we were asked, to evaluate whether we could say something about changing this occupational exposure limit values. Our finding is that we were not able, because there is no enough data, there is no enough studies which have been designed to really look at these combined exposures. So this document will be available. I think it will be public very soon. I think after summer holidays, sometime in, I think during the fall. But my point is to highlight that what you want to do, at least from the kind of, epidemiological point of view, that's very important. But again, I will emphasize that I think we have to also take into consideration the kind of, mechanistic studies. If we can include some mechanistic studies there, that will be, I think, in line with what IARC is doing for classification of carcinogens.
Yes, I agree, Dr. Naroui. I mean, what you're mentioning is very important. This combination and coupling of epi studies with a mechanistic approach to confirm or refute, let's say, the observational findings, it's very important. And also, these lower benzene exposures or Vitex exposures that we see now in the workplace, in Europe at least, it does not negate the fact. I mean, there is an interesting study that just came out from Vermeulen's group with on the supralinear, let's say, hematotoxicity profile of factory workers, where they could see how a nonlinear and inverse, let's say, nonlinear response of hematotoxicity against the benzene exposures was observed, which is quite interesting. I mean, this is worth looking more into it, and maybe in relation with a possible night shift work status that was not taken into account in their study. And maybe the night shift work status could explain, partially at least, some of the results obtained in their two factory sites that they have. I believe this is something that is worth looking into it. Okay, to better understand this the interplay, better say, between chemical benzene exposures and night shift work. And Joe wanted also to add something to it.
Yes, but you can. I can wait. You can finish your reasoning.
Please. Please
Yeah.
go ahead.
No, I just read the paper myself by Vermeulen and his group, and I think that's the slide that Besad showed at the end is where we, you know, is one of the former PhD students of Rule, Jelle Flanderen, when you look at the meta regression of benzene and the leukemia risk curve, and I think what we can do, I mean, there are of course limitations to what we can do with this cohort data. It's very coarse. So I totally agree with you, Sean. We need mechanistic data to better understand the plausibility of what we may observe on the, you know, on the higher level in terms of let's say we have the mechanistic high resolution data here, and we have the granular more coarse data up here. So we need to understand this together, and I think what we have been doing so far is just adding to one little piece of the IARC working group. It's the observational data. It's the etiological exposure risk analysis. But of course, there's a lot done on the mechanistic studies, and we should even do more. So I think I have actually one question for you, which I think is interesting, and what we saw in Leon's paper when we looked at rotating shift work and prostate cancer, we saw that rotating shift work was actually producing a higher hazard ratio than night shift work. So supporting the hypothesis that alternating between day and night is maybe worse than just staying in consecutive nights. Maybe we had few cases in the night shift category, so that also might be some statistical thing there. But there's a saying among the offshore workers that, so we have basically 4 categories. We have only day workers, only night workers. We have those who work 7 nights and swing to the day in the middle of the week, because everyone is working 14 days, 12 hours in 14 days. So it's quite extreme. Mm-hmm. And then we have the 4th category who is working night and swinging to day. Mm-hmm. So I think that many people, they don't want to swing from night to day. That's what they experience. That's the worst thing. Or I might misremember, but there's one of the swing shifts which is they don't want to do it. They prefer not to work that schedule, because the body, it's extremely difficult to go 7 nights and then swing to 7 days. Mm. They struggle a lot in the second part of their stay offshore. Mm. We don't have that data in the old cohort, but we have it in the new one. So my question to you is there any biological rationale for this? Is it this notion among the workers, could it be explained by some biological mechanism that it's actually worse to swing from night to day than day to night?
Well, I think it's very, you know, I mean, let's comment on the first thing, that it has kind of, you know, societal impact. I mean, how we really regulate, or what we recommend, and what type of shifts. And I'm not expert on that, but my colleague Jeniana, who has been working with that, an expert on that. So we are hearing that maybe kind of, you know, permanent, I mean, rotations are better than non-permanent. So regarding your question, I think, I don't know whether it has kind of, you know, biological impact, or it's mostly kind of, you know, how you call it, kind of, you know, non-biological stressor factors. So one thing is that because when we are talking about, especially, you know, these hydrocarbons, we should take into consideration 3 things. One is the metabolism, how body really regulates and metabolizes these hydrocarbons, and then how detoxify them, whether it's detoxification by these cytochrome P450 enzymes, or how they repair the damages. I mean, from the clinical studies, we know that the repair mechanisms, at least some of those who are central in chemotherapy, they have a kind of, circadian rhythmicity. And some of this, for drug therapies, they are recommended to be given at certain time of certain time of the whether night, where you have, maybe a higher activity of DNA repair. Again, we do not still understand how many of these, genes which are involved in the detoxification and repair and also metabolisms of these, metabolites are under control of the circadian rhythm pathway. So this is very, very relevant question, but I really do not know the answer before we really have done some studies, and that's the point. That is one of the questions that, I mean, one of the research needs that we have highlighted in the report, which is coming from the Nordic expert group, that we need really this type of, studies to do. And this is what's my point. We need really kind of, mechanistic studies to understand these things. We don't really understand. If you have, let's say, a worker who's working, 16 hours or 12 hours, is the person metabolizing, the compounds more, the metabolites are more metabolites in this person versus a person who's working, let's say, 8 hours, but other time points. So we don't know these things. So still, these are questions that I think is difficult to understand and difficult to answer, but we need this type of studies. That's my point. So I'm sorry, I cannot answer really your question.
Konstantinos?
Yes, yes, please. Yes, Michael.
Yes. Thank you for the invitation. You invite me in this conference. And I'm Michael Tornoridis. I'm working in epidemiology for 28 years now. And I can understand, and I understand how valuable are all these results we have today. Thank you very much for this. I have a question, which is for Dr. Roshan Nauri from Norway. He showed us a slide with the mode of action of chemical carcinogenesis. And he showed us the biological cycle of the mechanism happening when someone has cancer. He gave more emphasis on the outside cycle, but inside I have seen organic molecules, epoxides, benzene oxidation, and this kind of things. And because he tell us about mechanism and just a minute ago, I would like to ask if there are people who are working on this mechanism in his lab. The oxidation of the double bond to create and start the cancer. Are there people in the labs you are working who are interested in this subject and working on this subject? And what are the basic findings on this?
You just to answer your question. Yeah, we have been, working with lung cancer. So lung cancer is also a kind of, very complicated disease. And the first slide that I was showing, this multi-step carcinogenesis. And we have been also working with detoxification of different type of adducts in DNA. Apparently, we have also been looking at the, different, genetic susceptibility markers like, genetic polymorphisms in relation to lung cancer, breast cancer, and so on. And we have, yes, we have some publications there. We have been working with genomic instability as well, with DNA repair, and I've been looking, many of these factors. So recently we have been looking more on the, workers who are exposed to benzene, not, in the offshore, but benzene which may come out from this, diesel exhaust and so on. And then we have been looking at the epigenetic effects there and looking at the expression of these cytochrome P450 genes. Also, we have been looking at this, gender differences. So yes, we have done a number of mechanistic studies, both on benzene and also on the night shift workers.
Thank you.
And we are very much interested, in this topic, especially, this combined effect of chemical exposures, and it could be other exposures with night shift work. I'm talking about night shift work, and I think I pointed out because the last meeting of the IR, they had to specify. I was in one of these, in the monograph group, and they had to specify that we are talking about night shift work. We are not talking about, generally shift work, because if we think that one of the main mechanisms of carcinogenesis in shift workers is circadian disruption, then the circadian disruption happens when you have so-called, light at night pollution. Light during the dark, the night is considered a kind of pollution exposure. So that's the.
I was asking because in 1990, as a professor in Crete, I was working on this reaction with porphyrins, oxidize double bonds, and we tried to find the transition state or the intermediates. And I learned for the bibliography that this was the circle of the life. And so I think we can go on and try to find out what happened in the human body with this reaction in order to try to stop it, because carcinogenesis and atherosclerosis starting up from this reaction. We did some work on this, few steps, and we are interested in to go on with this particular reaction, much closer to the human body.
Thank you. Thank you for your comments and questions. I think they're very important. These mechanistic studies, yes, we're waiting for Dr. Naroui and others to actually lead these efforts, because it's very important to understand how the peripheral clocks and enzymes affected by them actually are influenced and how they're influencing chemical metabolism, hydrocarbon metabolism. We don't know this, particularly in human studies, and also we have some evidence from animal models, but we need more, definitely. Yes. Any other comments, questions?
One question to you, Konstantinos, and also Besad. You showed that there was the same atmospheric levels of benzene during night and day, correct?
Yes.
But still, you wait for the urine samples to be analyzed.
Yes.
So this is what you prefer to have. I mean, to have the equal between day and night is a good thing, because then if there is a difference in the urine taken at night, you know that you don't need to correct for the benzene intensity level, right?
Yes.
So my question is basically, you do expect, I guess, the hypothesis is that the urine levels will be showing higher biological potential from the benzene exposure at night. Is that correct?
I expect to see this chronodisrupted metabolism of benzene. Yes.
Yes. I see. So will you then employ omics platform to see this, or what is the main outcome of the analysis for the urine?
Yes, observing this chronodifferentiated, let's say, metabolism and time window, it's one thing. The other thing is to actually try to see what that means with respect to toxicity onset and disease process. And let's say, what is the net outcome of cell unit damage repair processes in the end. And this is the most interesting, I believe, fact. So you really need to follow this step by step, characterizing the external environment, moving on to the endogenous response, and then try to put this mechanistic or meet in the middle, let's say, biological layer of formation from systems biology to try to shed light on the possible differentiated risk or differentiated disease phenotype. If you deviate, let's say, from the average risk, you don't account for these timestamps and differentiated metabolism compared with a possibly stratified risk when you account for the night shift work, for example.
Thank you. Yes. No, I think it'd be really interesting to see the urine sample results. Look forward to that.
Yes, we're trying to analyze them, and also Dr. Eleftheriou is smiling, but because he is helping us also with the analysis. So we have to wait a little bit more for that.
Sure.
Okay, any other last comments or questions?
Maybe just one last comment.
Yes.
One of the things about night shift is that sleep is not just sleep disturbed, it's where in sleep is disturbed. And there are ways of, that's why I was excited coming in here, of looking at the structure of sleep in much more detail. And that has exact consequences about the things you're looking at. In other words, sleep is stratified, and the repair mechanism happening in specific parts, not in every part of sleep. And that also addresses the question that was raised earlier about the difficulty going back from night sleep to day sleep. If you pay a lot of effort to do something which you used to do for much of your life, and once you achieve it, it's extremely hard to go back to what you did before without having some consequences. So again, looking at sleep at those transitions, it would be really exciting in both understanding sleep and how we adjust to changes in the environment, and also for addressing some of the questions you have. One of the difficulties is that it's combining the chemical. It's very time-consuming experiments, as we find with Ligia, to do both. And for example, the group of people which I find most natural to do, because they have a lot to gain and it's in the interest of the companies, is kind of air pilots, because they are very special people. They have critical conditions, and one mistake they make, it makes all the difference. But looking at transitions, for example, in their schedule, it would be extremely useful, and I would be any such thing. It would be really exciting research to do for many ways, basic neuroscience to also applications. Thank you.
Yes, thank you, Dr. Anili. Very interesting also your study. Now you remind me, I had a question about your PCA analysis. I don't know who did the PCA. Should I direct the question to you or to Dr. Eleftheriou?
Yes, we did it. Basically, we have actually published a paper recently we have another grant from the research foundation, DECNO, in which we try to combine this kind of analysis, doing different applications, and we choose neuroscience and astrophysics, and it is exactly the same thing to do. It's basically standard. It's like PCA, but we also use Gaussian PCA, Gaussian kernel to do the analysis, which we did not have to use in this case because you know, everything works so nicely, surprisingly from the others. Like here was the one who did the analysis, who actually we found the results without making too much effort by putting the things together. So it's in a small way the kind of things that everybody's excited about AI. It's drawing a line to separate things using it in a multi-dimensional space.
Yes, so I wanted to ask you, the PCAs, you had two PCAs, if I remember correctly, one for each of the brain activity markers.
We can put into the PCA all of the frequency bands, all of the results from the chemistry analysis that then we can choose to restrict what to look. So it's a really powerful technique. We can put everything in, or we can actually bias it towards things which we expect to see, in which case it makes a lot of sense.
Yes, you had some blue points that seem to be clustered.
Oh, no.
What were these?
These blue points were the classification we made in advance, namely people who work in the pumps, people who were smokers. So we actually put the symbols separated. Okay, without doing any clustering, it's the classes were obviously separated by eye. You could see very clearly. So it's a very robust analysis. We did not have to make any assumptions and just label the things according to whether people worked on the pumps, worked in the offices, or there were controls, or there were smokers. Again, one thing which actually really surprised us, and I think I would mention it as an emphasis for doing things in a personal as well. There was one person in the petrol pumps, which just did not fit the thing. I think neither in terms of biochemistry, nor in terms of analysis. And we asked him, you know, why are you a Superman? Because the only thing we can find different is that he enjoys life. He has a very positive outlook. And I'm not telling this as a joke, because we know from your feedback that if you have a positive outlook, it does influence things, but that was really extreme.
Okay, thank you. Thank you all for your contributions. I believe it's time for lunch, more or less.
Especially in Cyprus. Thank you for organizing, Konstantinos.
Thank you all for your contributions. Very, very interesting. And we keep in touch.
Thank you.
Thank you so much for hosting, Konstantinos. Thank you.
Webinar.
Bye.
Thank you guys. Good to see you.
Okay. Bye-bye.
All right. Thank you.